Hey guys, Ricardo here, and today we're going to have a quick tutorial on cellular respiration. And more specifically, we're going to go over an overview of glycolysis, the Krebs cycle, and the electron transport chain. So let's just jump right into it. So, first of all, why do we do cellular respiration? Well, the basic purpose of cellular respiration is to convert glucose into energy. Now, our body needs energy to function, and so one of the major sources of this energy comes from this molecule, glucose. Now, this is a bit of an oversimplified formula. Maybe you've seen it in your biology textbook as glucose plus oxygen is converted to carbon dioxide, water, and energy. This is, for biologists at least, the equation of cellular respiration. And as we can see, we breathe in oxygen, this combines with the glucose, or at a sort of a derivative of glucose later on, we'll go into it, resulting in formation of carbon dioxide, which we dispel through breathing, water, which we also dispel through breathing, and energy, which allows us to keep on existing, which is very good. So, the stages of cellular respiration are as follows. So first of all, we have glycolysis. Glycolysis. Now, glycolysis is the first part of cellular respiration, and it results in the production of 2 ATP molecules. And it can occur with or without oxygen present. If oxygen is not present, so if no O2, it goes to anaerobic respiration, which we'll cover in a subsequent video. And well, that was shocking, anaerobic. If it does have oxygen present, we go into the next stage of cellular respiration, which is aerobic cellular respiration, and the first one of that is the Krebs, or the citric acid cycle. Now this also produces 2 ATPs per molecule of glucose, and then following this, a lot of the other products in the Krebs cycle reaction go on to the electron transport chain. Electron transport chain. And this can produce up to, this can produce up to 34 ATP. So this is the high-end yield of the cellular respiration process. And it's important to note that this overall figure of 38 ATP is a theoretical maximum. There's still lots of research going on to exactly identify where this lies, but there is a lot of variability, and it can go from as low as 29 ATP all the way up to this maximum of 38 ATP. And apart from that, we're going to jump straight into it. And so, glycolysis. First of all, let's look at the meaning of the word itself. Glyco, it's glucose, lysis, it's splitting. So what we're basically doing in this stage is splitting glucose. Quite self-explanatory. Now, glucose is a 6-carbon molecule. 4, 5, 6. So you can see 1, 2, 3, 4, 5, 6. And we split it into 2 3-carbon molecules. Makes sense? These 3 carbon molecules are called pyruvate. Now, this all happens in the cytosol of the cell. So if we draw a cell real quick, we have our nucleus, and we have all our organelles, we have our mitochondria, and so some glucose gets in and goes through glycolysis in the cytosol. This can occur with and without oxygen, as mentioned before, but it only produces the small amount of 2 ATP. However, it's important to mention that during this phase, at the beginning, there is a phase called the energy investment phase to glycolysis. Energy investment phase. And in this phase, 2 ATP are actually used up. And now this is used to get the sort of glucose molecule into sort of an altered higher energy state, where it can then be broken down in the energy payoff phase. And the energy payoff phase results in the generation of 4 ATP. So in total, we get 2 ATP, but it's important to remember that 2 ATP are initially invested to get the glucose molecule up to a higher energy state, and then that higher energy glucose state is broken down to 2 3-carbon molecules called pyruvate to generate 4 ATP in total, which results in the net production of 2 ATP. Now, this is the sort of critical junction between anaerobic and aerobic respiration. If oxygen is present, the pyruvate molecule will then go into the mitochondria, where we'll explain what happens next. However, if no oxygen is present, it will undergo anaerobic respiration, which depending on the organism could be fermentation or lactic acid formation, but we'll go into that in another video. So if oxygen is present, we go into the mitochondria. And we're going to explore that now. Now, once the pyruvate gets into the mitochondria, it immediately gets converted to acetyl-CoA. So we have this 3-carbon molecule of pyruvate, and it gets converted into acetyl-CoA, which is a 2-carbon molecule with a little group which we'll call CoA here. Now, the cleaving of this carbon, as we can see, we go from a 3-carbon molecule to a 2-carbon backbone molecule, results in the production of CO2, so that's where that other carbon goes. It goes into the CO2, and also results in the conversion of NAD+ to NADH. So that gets converted to NADH. And so now, why do we convert it to acetyl-CoA? Well, acetyl-CoA is the molecule which is required to enter the citric acid or Krebs cycle. Now what happens is the acetyl-CoA fuses with oxaloacetic acid, the name of which is not important unless you're doing a college level course. So basically it fuses with some sort of molecule. So we got this, and it fuses with some sort of molecule. And then it goes through a whole bunch of reactions, which end up in the reproduction of this oxaloacetic acid. So then the next molecule of acetyl-CoA can come in and the cycle can repeat. That's why it's called a cycle, because the substrate here is regenerated at the end of the series of reactions. Now for each sort of round of one molecule of acetyl-CoA, 1 ATP is produced, 3 NADHs are produced, 1 FADH2, 3 hydrogen ions, and as well as 2 carbon dioxides. All of this happens, as we said before, inside the mitochondria, and more specifically inside the mitochondrial matrix. And so I think an important question to ask is, why do we create all these molecules? I mean, we've only produced 1 ATP, which is great, but we've got lots of these other strange molecules like NADH and FADH2. It's important to note that these molecules are basically energy carriers. So they basically store a lot of energy, a lot more than 1 ATP, and this energy is going to be sort of harvested in the next phase of cellular respiration, the electron transport chain, to generate ATP. So these are like energy stores, which are going to be rewarded later on. And so just to summarize this phase, it requires oxygen, so oxygen is needed, and it happens in the mitochondrial matrix. Basically, pyruvate is initially converted to acetyl-CoA, cleaving off 1 carbon, which becomes carbon dioxide, and creating 1 molecule of NADH. The acetyl-CoA then enters this cycle of a series of reactions, which regenerates the same product at the end. And each molecule of acetyl-CoA produces 1 ATP, 3 NADH, 1 FADH2, and 3 hydrogen ions. It's important to remember that 1 molecule of glucose, so 1 glucose, results in 2 pyruvate, which results in 2 acetyl-CoAs. So really, for each molecule of glucose, we produce 2 ATP in this phase, we produce 6 NADHs, 2 FADH2s, and 6 hydrogen ions. Alright, moving on to the next stage. The next stage is the electron transport chain. It is the highest yielding part, energy-wise, in the process of cellular respiration, and it occurs at the inner mitochondrial membrane. So here we have our phospholipid bilayer, which is on the inside of the mitochondria. And we have all these proteins along here. And we have one special protein at the end called ATP synthase. Now what's going to happen is all the molecules which have been created in the Krebs cycle and glycolysis, such as FADH, are going to come here, undergo a special reaction, and regenerate the original FAD+. In doing this, electrons are going to be stored here, and a hydrogen proton is going to be expelled. So the hydrogen goes outside, and the electrons are stored here. The electrons move between these proteins, undergoing a series of complex reactions. And these electrons allow for the further splitting of NADH, sorry, that should be FADH2, NADHs to NAD pluses, and etc. And they're going to continue pumping out the protons. So basically these high energy molecules are releasing their energy and pumping out protons across the inner mitochondrial membrane. And so we end up with a lot of hydrogen atoms on the outside. Now, what happens next is sort of this process called chemiosmosis. Now, this word basically means producing energy from chemical gradients. And so here we've created a chemical gradient. We have a higher concentration of hydrogen ions outside the membrane, the inner mitochondrial membrane, than inside the inner mitochondrial membrane. So what's going to happen is these hydrogen ions are going to go flow down their concentration gradient, going through this molecule, this protein rather, of ATP synthase. Now, the name is pretty self-explanatory. ATP, it's the molecule, and synthase is what it is making of. So it's going to make ATP. So basically, as these hydrogen ions all flow through the ATP synthase, it causes a series of reactions which result in the generation of ATP, up to 34 ATP in total for every molecule of glucose. And so this sort of set of reactions require oxygen, because in this final one here, oxygen is combined with hydrogen to form water, and is expelled outside until we breathe it out. And so that's how we get rid of the electrons here. The electrons combine, resulting in the formation of water. And it, as we said before, produces up to 34 ATP. It works along the inner mitochondrial membrane, works by this process called chemiosmosis, which is the generating of energy via electrochemical gradient. And so we have all these high energy molecules which have been created in the Krebs cycle and glycolysis, sort of bringing their electrons here into these proteins along the membrane. These electrons also help in the pumping out of the hydrogen ions, and this creates a gradient which generates ATP through this molecule ATP synthase. So let's do a little summary flowchart of what we've covered. We start off with glycolysis, Glycolysis. Now, if no oxygen, we go anaerobic. If oxygen, we go to the Kreb cycle. Kreb cycle then leads to the electron transport chain. And so we have 2 ATPs per molecule of glucose here, because so remember, glucose is split out into pyruvate. Pyruvate, and this happens in the cytosol. The pyruvate then goes across the membrane of the mitochondria and undergoes a change to become acetyl-CoA. And remember, that was the cleaving off of the carbon to form that. And so here we then generate overall for each molecule of glucose, remember, so that's for 2 molecules of acetyl-CoA. We generate another further 2 ATP. And then in the electron transport chain, we have all those high energy electron carriers reacting and pumping out those protons to form a chemical gradient, resulting in a theoretical maximum of 34 ATPs. All things prior to this line require oxygen. Things after this line, so rather all things before this line don't require oxygen, so can occur without oxygen. All things after require oxygen, of course, apart from the anaerobic, which is different. We'll cover that in a different video. And also these all occur in the mitochondria, while glycolysis occurs in the cytosol. And so that's a rough overview of cellular respiration. I know it was a bit rushed, a bit quick, but I hope I got across all the key information as quickly as possible. On the next page I have a bunch of questions which hopefully you'll be able to answer after watching this video. Just go to them now. And so I'll put the answers to this in the description. Also let me know if there's any ways I can improve these videos, anything you want me to cover in future videos, or just any tips and life facts in general. Thanks so much guys, I really appreciate your time if you stuck through, and happy studying.